Today, we are going to continue talking about protein synthesis. We've previously talked about step one of protein synthesis, which is called transcription. During that phase, all that's happening is we're taking that DNA molecule, we're opening it up, and we're reading only one side of the DNA, and we're just going to rewrite that into messenger RNA. And when that happens, remember, we're going from the A's, G's, C's, and T's to A's, G's, C's, and U's. Once that messenger RNA has been created, it will then leave the nucleus and it'll go out into the gooey cytoplasm of the cell. It'll go find a ribosome where it'll then be read to create a protein molecule. And that step that's happening at the ribosome, that is translation, and that's what we're going to talk about today. So, translation is happening at the ribosome. And what's happening here is that a ribosome is actually going to read all of these letters, the A's, G's, C's, and U's, that are making up this messenger RNA strand. And as the ribosome is reading these letters, it's going to read it in chunks of 3. So every set of 3 letters you see here in this diagram, that's what we call a codon. And so we see GAG, CCC, UCA. Each of these sets of 3 letters is called a codon. And that's how the ribosome is actually going to read this messenger RNA. So if we look at this diagram here, it says how many codons are in the messenger RNA molecule above. So you could just count these up. Every 3 of these sets of letters or bases, that's going to be a codon. And so if I counted those, I would say there's 7 codons there. And each codon is made up of 3 bases. And it's either going to be an A, a C, a G, or a U, since this is the RNA language. There are no T's here. So as the ribosome reads the messenger RNA codons, transfer RNA molecules are going to come in and match up what we call an anticodon to the codons that are on the mRNA. So let's see what's happening here. So there's our transfer RNA, kind of has that funky shape there. But on the bottom side of it, you'll see 3 other letters, and those are RNA nucleotides. They are bases that are a part of the RNA molecule. And so what ends up happening is that those 3 letters on the bottom of the transfer RNA are going to match up with the 3 letters that are on the mRNA. So the 3 letters on the messenger RNA are called the codon, and the 3 letters on the transfer RNA are called the anticodon, and they're going to be complementary. So right here, we see that the anticodon that's on the bottom of the tRNA is CUC. And that matches up with the codon we see on the messenger RNA, which is GAG. They match up. So apples grow on trees and cars go in garages. But remember, since this is RNA here, we don't have a T, we have a U instead. The thing that's so important about the transfer RNA is that sticking on the top of it, we see that big circle there, that's representing an amino acid. And remember that the amino acids are the monomers of protein. And that's what we're making here. That's the whole point of protein synthesis, is to actually create protein molecules. And so in order to make a protein, we have to have the monomers to do so. And those monomers are called amino acids. And so the transfer RNA, that's what's going to be bringing in those amino acids to this translation step. And so as the transfer molecules or the transfer RNA molecules are coming in, they're just going to continue matching up their anticodons with the codons, and each tRNA molecule will have a different amino acid attached to the top of it. So we have that purple one that's getting added in first, and now I see a brown amino acid that's getting added in next. And that tRNA is matching up its anticodon with the codon. So what would that anticodon be? It would be GGG, because that's complementary to the codon down there, which says CCC. So they'll just keep matching themselves up that way. And as the amino acids get brought in, they're going to stick together by using peptide bonds. And so the amino acids keep getting carried in on the tops of the transfer RNA molecules. They'll get joined together by peptide bonds, and that is what is creating the protein molecule. So that's what you see happening here. We now have a third amino acid getting brought in on the top of that transfer RNA molecule, has the anticodon AGU on the bottom, matching up with the codon UCA, which is on the messenger RNA. And so now our growing protein molecule has 3 amino acids in it. And there are actually 20 different types of amino acids. And there is a way to determine the exact amino acids that those are by using something called a messenger RNA codon chart. So you can actually look up the codons in this chart to figure out what amino acid is stuck to the top of that transfer RNA. So you can figure out the exact amino acids that are getting added into that growing protein molecule. So this says the amino acids that are added to the protein are based on the codon that the tRNA is matched up with. So on this slide here, we see this crazy looking chart on the right. It's got a lot of letters. It's got a lot of amino acid abbreviations on the inside of it. And we can use that chart to figure out the amino acids that are going to get added into this protein molecule. And so for this example, it looks like our codon is UCA. And so I can look up that codon in this chart I see on the right of the screen here. And I'm just going to look it up one letter at a time. So the first letter is a U. So on the left-hand side of this table, I'm going to find a U, which just so happens to be at the top, that first section. And so my answer, my amino acid, is going to be in that section. The second letter in this codon is a C. So across the top, I find the C column, and that's going to be right here. Where these boxes overlap, that's where my answer is going to be. The third letter in the codon is an A. And so on the right-hand side of the table, I find the row that has the A, which is the third one down. And if I find the place where all 3 of those sections overlap, that's my answer. And so ser, which is serine, that is the amino acid that is stuck to the top of that transfer RNA molecule. Okay, and I was able to figure that out by looking up the codon in that codon chart. Okay, so we're going to practice this a couple more times. So this says, for the following DNA examples, first give the mRNA sequence, then use the mRNA codon chart to determine the amino acid that'll be added into the protein molecule. So we're given just a very small DNA section here, it's just TAC. We're going to first transcribe that into messenger RNA. So that's going to be complementary, but I'm not going to use T's. I'm going to use a U instead. So if the DNA is TAC, then my messenger RNA is going to be AUG. And once I have that, I can look that up in the table here. So the first letter I find on the left, which is going to be the third section down because that's the A. And then the U, I find that across the top. That's the first column. And where those sections overlap, that's where my answer is going to be. And then the final letter is a G. And so that looks like it's that bottom row there in that box. And so my answer is going to be MET, which is methionine. So these are all just abbreviations in the table, but the MET stands for the amino acid methionine. You don't need to memorize those, but that's what that stands for. Okay, second example, GTT. So if we transcribe that into messenger RNA, that's going to be CAA. We look that up in the table. Okay, so the C is the first letter. I find that on the left, so that's the section I'm in. The A, I find that across the top. And then the third letter for that codon is an A, which means the place where all 3 of those sections overlap is going to be GLN, and that stands for glutamine. So that is that amino acid. Okay, final example here. So we have ATT as our DNA. So we're going to first transcribe that into the messenger RNA language, which would be UAA. Look that up in the table. So we got the U, the A, the third letter is an A, and this one's a little bit different. When I find that answer, it actually says stop. which is not a weird name for an amino acid. That is just like a stop message to tell the ribosome, hey, you're done. You made the protein. Good job. And then it knows that it can release the protein. It's been fully created and it finished its job. Okay, so just to wrap all of this up, remember that protein synthesis has two steps. And the steps actually are in alphabetical order. So transcription comes first. What's happening here is that we're in the nucleus, we take the DNA, and we're just going to rewrite it into that messenger RNA language. We do not use T's, we use U's instead. Once that is created, the messenger RNA leaves the nucleus where it can go out and find a ribosome, and the ribosome will read that messenger RNA in sets of 3 letters. Each set of 3 bases is called a codon. And we use those codons and look them up in the mRNA codon chart to figure out what exact amino acids are getting brought in on the tops of the transfer RNA to be put together to build a protein. So the amino acids get joined together by peptide bonds to form a protein molecule. And again, all of this is protein synthesis. So it's 2 steps. And the end game here, the whole point of this is to use the DNA that has the directions to make proteins to actually make a protein molecule at the ribosome inside of your cells. And that is protein synthesis.